to be here in Italy. I enjoyed very much. I was asked to speak about some of the new biomarkers, and as a result, I think we've learned some new concepts related to acute kidney injury that flow from these markers. So I'll not only speak about the markers, but I'll speak about some new concepts in acute kidney injury. My conflict of interest disclosures are there. I am going to talk about, in particular, some markers made by Astute Medical. So let's begin with sort of the revised conceptual model for acute kidney injury. And I say revised because it's sort of a model that's continuing to evolve as we understand this disease process better. This is a model that recognizes that things are changing. It's a dynamic process. There may be an evolving acute kidney injury, but there's also recovery, and this is important as well, and we hope that, of course, patients can get back to baseline. This also integrates various biomarkers of damage as well as functional markers like serum creatinine and cystatin C. Urine output, as we've discussed, is a bit of a functional marker as well, although certainly a non-specific one, although a bit more sensitive than serum creatinine for rapid changes in GFR. The problem is that the relationship between functional change and damage is not necessarily a simplistic one. So in this slide, you have on the very top, panel A shows that damage is actually starting before there's a change in function. And this is what Stefano talked about in terms of functional renal reserve, right? You can have loss of nephrons from an injurious process in a patient who has normal renal function, and there's no change in function until you reach a certain threshold where enough decrease in nephrons, or at least functioning nephrons, to cause the creatinine to change. Or you can have a situation where functional change and damage are occurring simultaneously, which is in the middle, or a situation such as what we might refer to as a pre-renal condition, which leads to damage as the hypoperfusion becomes worse and worse, where it begins early on with a purely functional change that then results in damage. And then underlying all of this is the fact that the kidney's not just sitting there and ignoring this process, but that the kidney is under stress while these processes occur, and perhaps we can detect that stress. So let me introduce this concept of kidney stress. So kidney stress is a response to a non-lethal cellular injury, or even just a noxious stimuli that is not injurious per se. It may include a protective response pattern or a downregulation of non-vital cellular function. So this decrease in function is perhaps at the expense of overall organ function, but is a protective mechanism in terms of cellular bioenergetics. This slide shows the concepts related to sepsis-associated AKI, but it turns out that surgery-associated AKI is very similar. In sepsis, you have pattern-associated molecular patterns from the pathogen itself, such as endotoxin or various other bacterial products which are being filtered at the glomerulus and also potentially delivered to the kidney through the peritubular capillary. In a surgical situation, you may have damage-associated molecular patterns, which actually signal through the same toll-like receptors in the proximal tubule and cause inflammation to occur in the kidney. The kidney signals inside the epithelial cells, that the proximal tubule epithelial cells, which first see this insult, signal themselves and other neighboring cells distal in the nephron that something has happened, that they're in fact sensing a injurious stimuli or pathogenic stimuli somewhere in the body, which is being then filtered and then concentrated in the proximal tubule effluent. It's interesting that this system is designed not really to detect retrograde infection, because the distal tubule cells don't have nearly as capable apparatus to detect this signature than the proximal tubule. So it's really designed to detect molecules coming across the glomerulus, and these pathogen-associated, these pattern recognition receptors, G-couple receptors, many of which are even olfactory and taste receptor like. And so literally the kidney smells and tastes what's coming across the glomerulus and is warned that there is noxious stimuli coming from somewhere in the body. And as a response to this, it downregulates the proximal tubule epithelial cells as well as distal tubule epithelial cells, downregulate their response, and within minutes, or at least within a few hours of cecal ligation and puncture, for example, there is a massive downregulation of ATP in whole tissue isolates, and there's evidence of decreased energy utilization in cells as well. Several biomarkers increase, some of which increase already before there is any evidence of injury to the kidney by histology or molecular changes. And plasma NGAL in particular seems to be a very early marker, probably more of a marker of the systemic inflammation than it is a marker of kidney injury. Urine NGAL is a mixture of what's coming across in terms of filtration as well as what's being produced by the kidney itself, and therefore is more of an injury marker. But there are emergence of new markers in the last few years, tissue inhibitor metalloproteases 2 and insulin-like growth factor binding protein 7. These markers are markers of cell cycle arrest, and they were more predictive of acute kidney injury than any of the existing biomarkers. Importantly, this concept of cell cycle arrest is relevant to this discussion because the process of cell cycle arrest is a protective mechanism. Epithelial cells throughout the body respond to environmental stimuli, noxious environmental stimuli, through a variety of protective mechanisms, one of which is to get out of cell cycle if the DNA might be damaged. So epithelial cells behave. I tell my children, I say, reproduction is dangerous and expensive, so don't do it unless you're ready. And epithelial cells take the same response. If it's dangerous because if the DNA is damaged, the daughter cells will apoptose. It's also the most bioenergetically expensive thing a cell can do is to reproduce all of its DNA in preparation for cell division. So it doesn't want to do that if it's in an environment that's hostile. And so even renal tube epithelial cells respond to stimuli like UV radiation because they're epithelial in nature, despite the fact they haven't seen UV radiation in billions of years, or millions of years anyway, of evolution. So this is a complicated slide. I don't want to go through all of it, but just if you can pay attention to the upper left-hand panel and the lower right-hand panel. In the lower right-hand panel, I'm showing you both distal and proximal tubule epithelial cells in culture. These are primary culture taken from human donor cells. Organs that were discarded for transplantation were then denatured, and then we culture the tubular cells. And so the control is C, so no change. The N is nutrient deprivation, and O is oxygen and nutrient deprivation. And you can see just with nutrient deprivation, which is not, does not cause the LDH to go up, so there's not a damage to the cell. The cells are alive. They're not dying. Yet, as you can see from the upper, and that's true for both proximal and tubular and distal tubular cells, they're not, it's a noxious stimuli, but it's not an injurious stimuli. And you can see just from that noxious stimuli from the upper left-hand panel, you can see that these conditions cause a burst of biomarker release from the cells. And so it's a stress signal, not a damage signal. So you have to think of it differently than a troponin for the kidney. It's not that. It's a stress marker, not a damage marker. And here you can see it's different than the other biomarkers that are markers of injury. You can see that IGF-BP7, for example, co-localizes. See if I have a pointer here. If it co-localizes with KIM1 in some tubular cells, but not others. Similarly, it has very little co-localization with NGAL. TIMP2 has a little bit of co-localization with KIM1, but much more co-localization with NGAL, but there are still cells that are positive for KIM1, or sorry, positive for TIMP2, but not positive for NGAL. And of course, you can understand how that would work, right? You could have a damaged cell, which is no longer under stress because the noxious stimuli has gone away, so it's positive for NGAL, but negative for TIM2, or it's a cell that could be positive for TIM2 because there's stress, but there's no damage yet, so it's negative for NGAL, or you could have a cell that has both damage and still ongoing stress. This has also been seen in a study that Melanie and Alex Zarbach and others conducted a few years ago, in which they took patients to the OR and did remote ischemic preconditioning as a way of trying to stimulate this protective response. And what they found was that by doing remote ischemic preconditioning, putting a blood pressure cuff on the arm, releasing damage-associated molecular patterns in low concentration into the plasma, HMGB1, uric acid, myoglobin, these molecules go to the kidney, are filtered at the glomerulus, and then stimulate those processes inside the proximal tube epithelial cell. And you can see that remote ischemic preconditioning increased TIMP2 and IGF-BP7 and then subsequently protected the kidney, whereas there was no change in NGAL until you actually did the cardiac surgery, which then made the NGAL go up. In high-risk patients, this seems to be, although in low-risk patients it doesn't seem to be protective, in high-risk patients, this process of stimulating TIMP2 and IGF-BP7 in this study was shown to be protective of subsequent damage from the surgery itself. And of course, using these biomarkers so it's one thing if you stimulate them to go off, but if this alarm goes off by itself, it tells you that the kidney thinks, to the extent that a kidney thinks, it's under stress. And so the way you think of this as a clinician is to say, this kidney thinks it's under stress. Why is that? And have I relieved that stress? Or is there still something I need to do? And you can see how this would map potentially to the KDIGO guideline. And I can think about discontinuing nephrotoxic drugs. I can think about ensuring adequate fluid status. And that, as we've just discussed already, that's not just giving fluid, but it's monitoring what I'm giving so that I'm avoiding both underfilling and overload, et cetera, et cetera. This is the response to vancomycin administration. So you can see patients that develop kidney injury. This is their biomarker signature around the dose of vancomycin, whereas if patients that don't develop kidney injury or just develop AKI stage 1 don't have an increase in biomarkers. We use this clinically when we add nephrotoxic agents to patients, and you do use a lot of nephrotoxic drugs in critically ill patients. You use a lot of nonsteroidals, you use a lot of calcineurin inhibitors in transplant patients, you use a lot of antibiotics, many of which have nephrotoxic potential, and as we add these drugs to patients, we monitor their stress. And as their kidney stress changes, we can anticipate AKI. Now, you can't always stop the nephrotoxic drugs, but you can dose them better, and you can avoid unnecessary nephrotoxic drugs. It's arguably no patient really needs a nonsteroidal, although we have an opioid epidemic, and so we want to use nonsteroidals in patients that don't have a high risk for acute kidney injury. This has been nicely showed. Many people have shown this slide already. This is work from Melanie Mersch and colleagues in Munster, in which the use of these biomarkers has been applied for identifying high-risk patients. About a third to 40% of patients are at high risk. These patients are then subjected to a very careful fluid management protocol guided by not just fluid management, but also inotropes and vasopressors guided by stroke volume variation, or whatever your technique of preference is in terms of functional hemodynamic monitoring, as well as the rest of the KDIGO bundle, and you can see this decreases the rate of acute kidney injury. This is important for a variety of reasons. One is it tells us that AKI is not inevitable in our perioperative patients, or any patient for that matter. There are many things that we can do to avoid acute kidney injury. But if we apply these unselectively to all patients, it means that we're essentially applying unnecessary treatments to 2/3 of our patients, which will not benefit them and will only drive up costs, delay discharge, et cetera. So we need to be selective about who we apply these measures to, and one way to do that is to use these new biomarkers. Lastly, I'll just show you this slide, which isn't published yet. This is an analysis that is still being completed for publication, but it nicely shows the pattern of multiple measurements. So these are measurements that were done 12 hours apart, and you can see that if you have two measures of TIMP2, IGF-BP7 that are both above 2, it's really diagnostic for acute kidney injury. This is basically has a positive predictive value that begins to approach 100%. If, on the other hand, you have one value that's above 2 and the next value is coming down, the risk is still high, but it's dramatically lower than it was if they both remain elevated. Now, this study was done without clinicians knowing the result of the first or the second measure. So it's conceivable that if you know the result of the first value is high, and you say, oh, the kidney's under a great deal of stress, can I alleviate that stress in some way? Perhaps by giving a fluid bolus if the patient needs that, perhaps by reducing, improving right heart dysfunction if it's in fact the other way around, or taking away a nephrotoxic drug. 12 hours later, if that kidney is still under stress, they're going to develop acute kidney injury almost inevitably. But if you've managed to do something which results in a decrease in their biomarker concentration within the next 12 hours, their rate of AKI is dramatically lower. Equally, or conversely, if you're negative, if you're below even the low cutoff, if you're below 0.3, you're protected for the next 12 hours, but only the next 12 hours because other things happen. And a patient develops a new exposure in 12 hours, the biomarker can't predict that. But if you continue to have negative values, in fact, it's not shown here, but if you have about 3 negative values in a row, 12 hours apart, the risk of AKI in that population is extremely low. So you can use these tests, therefore, in a dynamic fashion to understand whether or not the patient's at very high risk for the next 12 hours, or in the cases of continued stress, whether the patient's actually ruling in for acute kidney injury. And when I say acute kidney injury in these studies, I'm talking about stage 2 or 3 AKI, so it's really rather severe acute kidney injury. So in conclusion, renal stress response occurs even in non-lethal cell injury or noxious stimuli. It may include a protective response pattern or downregulation of non-vital cell functions. Markers of cell cycle arrest appear to be robust measures of kidney stress rather than damage per se, and that makes them different from other biomarkers. Underlying biology suggests an alarm phase marker before actual damage has occurred. There is a window of action before stress results in injury in most patients. Of course, you don't always know that, because if you make a measurement in a patient who just come in through the emergency department, for example, you don't know whether the injury's actually already occurred, and you've missed the window. But certainly in a perioperative patient that you've monitored through the case and into the perioperative period, you have that potential much more. Persistent stress becomes diagnostic of AKI and identifies risks for worse outcomes. And with that, I thank you for your attention.
Thank you very much, Professor Kellum.